Hello, I'm Bretton, one of our MCAT tutors here at Inspira Advantage, where we help students get into med school and other professional programs. In this video, we will learn about the 4 levels of protein structure: primary, secondary, tertiary, and quaternary. These levels describe the organization of amino acid chains in a protein molecule to varying levels of complexity and interactivity. Let's start with the most simple forms and work our way up. Beginning with primary protein structure. The primary structure of a protein is simply the linear sequence of amino acids joined by those peptide bonds. The sequence is determined by the genetic code in DNA, and even a single change in the sequence can affect the function of the protein. Primary structure is crucial for protein folding and the formation of higher order structures. In other words, if primary structure is lost, everything's going to be lost. Several factors can interfere with the primary structure of a protein. For example, errors during transcription from DNA or translation from RNA can lead to mutations in the amino acid sequence. Mutations can be caused by a variety of factors, such as exposure to mutagens, radiation, or other chemical agents. These mutations can result in misfolding or improper folding of the protein, which can then cause the protein to lose its biological activity or even become toxic to the cell, like in the case of Alzheimer's. In addition to mutations, post-translational modifications can also interfere with primary structure. Post-translational modifications include chemical modifications that occur after translation, such as phosphorylation, acetylation, or glycosylation. These modifications can alter the chemical properties of amino acids and affect the folding or function of a protein. The secondary structure of a protein refers to the local folding patterns that arise due to interactions between nearby amino acids. The most common secondary structures are the alpha helix and beta sheet. Alpha helices, shown here in red, are spiral structures formed by hydrogen bonding between amino acids within the same chain. Beta sheets, however, are planar structures formed by hydrogen bonding between amino acids in different chains. Secondary structures are important for determining the overall shape of the protein. For the MCAT, beta sheet and alpha helix are pretty synonymous with secondary structure. Well, how can we break this? Certain factors can interfere with the hydrogen bonding and disrupt the secondary structure of a protein. One such factor is high temperatures, which can break the hydrogen bonds and cause the protein to lose its shape. Another factor is pH, or ionic strength, of the surrounding environment, which can also interfere with the hydrogen bonding and cause the protein to lose its secondary structure. And this would make sense. We know that pH is just the concentration of hydrogens in solution. And if we have a bunch of hydrogens in solution, depending on the pKa of a certain R group, maybe it'll be protonated when it shouldn't be, or deprotonated when it shouldn't be. This is going to really mess up those intermolecular interactions we were talking about in our last video. Next, things are starting to get interesting when we move down to tertiary protein structure. The tertiary structure of a protein refers to the three-dimensional folding of an entire protein molecule. The folding is determined by the interactions between distant amino acids, such as hydrogen bonding, disulfide bridges, hydrophobic interactions, and electrostatic interactions. The tertiary structure is critical for the function of a protein and is responsible for its specific binding to other molecules. The tertiary structure of a protein is held together by a variety of interactions between amino acids. Any factor that disrupts these interactions can interfere with the tertiary structures of the protein. For example, again, we see high temperature coming up, changes in pH, or in ionic strength of the surrounding solution. This all makes sense. If we're messing with our secondary structure, we would predict that the tertiary structure then would as well be affected. What we see is different is now, I say, reducing sulfur-sulfur bonds. If we expose the protein to a reducing agent, it will break these bonds and cause the protein to lose the tertiary structure. This can have serious consequences for all the protein's function, as many proteins require their specific three-dimensional structure to actually perform their important biological roles. Notably, when we are running a denaturing SDS gel, we are destroying the tertiary structure of a protein to make it run based on its primary structure alone. And finally, on to quaternary structure. The quaternary structure of a protein refers to the arrangement of multiple protein subunits into a single functional unit. Each subunit may have its own primary, secondary, and tertiary structure, and the final structure of the protein is determined by the interactions between these subunits. The quaternary structure is important for the function of many proteins, including enzymes, antibodies, and one you might be familiar with, hemoglobin. And to make this really clear, quaternary structures are made up of different strands of amino acids. So these are all coded at different parts on the genome, and they'll come together and form this super complex of protein. As we said, the quaternary structure of a protein is the arrangement of multiple protein subunits into a single functional unit. Therefore, any factor that interferes with the interactions between these subunits can disrupt the quaternary structure of the protein. For example, changes in pH or ionic strength can disrupt the interactions between the subunits, as can exposure to certain chemicals or detergents, like SES. In addition, mutations in the genes that encode these subunits can lead to changes in the amino acid sequence, which can then affect the secondary structure, then the tertiary, and therefore the quaternary structure. I think you're seeing what I'm getting at here. We've got a domino effect. If we mess with primary, everything after that is going to be messed with as well, and we're going to have a very non-functional protein. This can have serious consequences for the protein's function and their ability to perform its biological roles. In summary, the 4 levels of protein structure, primary, secondary, tertiary, and quaternary, describe the organization of amino acid chains in a protein molecule. Understanding these levels is incredibly important for understanding the structure and function of proteins and biological enzymes, as well as getting a lot of questions right on the MCAT. So thank you so much for watching our video on protein structure, and I will see you next time.